Hello, everybody, and welcome to our class where we are going to start analyzing 483 consensus peaks that we've generated. In a previous 06 run.sh, we had all the design file for the entire class, and we've run that to run consensus peaks in a previous video. And we stopped with just a few running, but this is already pre-cooked. If you're running this from home, I don't recommend running all of these. I might pick a subset and you can follow along with the rest of the course with all of these, but it'll take a long time to run, and you have to watch it and make sure there's no errors and restart it. So that took about 4 days to run. So if you're taking this class, or I can make these available, we have here our consensus peaks, where we use our make consensus peaks function to merge all replicates of a given DNA binding protein's Chip-seq replicates into one final consensus peak using create consensus peaks function in our util folder. And so we have them all in here. We have the libraries we're going to need for today loaded, and I went ahead and loaded GenCode GR. If you want to do that, it's around line 150 something, I believe, where we're going to load it, and it'll take a little while to load. So go ahead and do that now if you want, and it can be running in the meantime. Okay, so let's start. So we have all these consensus peaks in a folder in results consensus peaks here. And what we can do is list files. Again, we kind of did this in the create consensus peak video, but we're going to list the files that are in there. We're just simply going to get their file names, and we're going to tell it where to go and that we want the full name of the file. So it's going to go into results, consensus peaks, and grab this file name here. Okay, so then the next thing we're going to do is an lapply. So list apply always wants to tell you what you're working on and then what the function is. And what's cool about that function is it can handle running a function from a different package. Even though lapply is base R, it's calling the function rtrack layer import, which we've seen before. We'll take these bed files and make them into G ranges. And that way we can do intersecting and a bunch of plotting that we'll do today. Okay, so let's make this. This may take a second as well to run this part here. It's got to go make all those file names. Once it does that, what we'll do is we'll just look at the 40-second entry. You can pick any entry you want just to see what this is going to sort of look like when we get the output here. So we'll look into consensus peaks, which we just made, into the 40-second index and its name. And then we'll pipe that to head. And what we'll see is the name of the 40-second entry is this protein DMTF1, and each of its peaks are numbered 1 through however long this is. We just piped it to head. And so what we want to do, if we look at what we just made, is this consensus peak list of G ranges. So there's 483 G ranges, each G range containing all the peaks of that given chip experiment. But we can see there's no name here. And we're going to want to know what the name of the DNA binding protein is before we move forward. So we'll use the names function to name them. So now we can see how useful this names function is. We're just going to have it go into consensus peak and set a name. That name will come from sapply, which will work on consensus peaks, the object we made up here. And it's going to run the function, a generic function x, which is just going to unlist string split, which is a common combo here. But the string split is going to split on the name in the entry that it's running. And it's going to break this underscore, which we have here. So basically it's going to read DMTF1 underscore 1. And what string split is going to do is say, take out let's split it at this underscore. So now we're going to have two halves, what's on the left half of the underscore and what's on the right half. And what we're telling it here is we just want the right half, the left half, sorry. So we just want to get this name. You could do this many different ways. This is just one way of saying, okay, I know the name is in the peak. The peaks are named by the DNA binding protein. I just want to get the name of it and not the peak number. And so that's what we will do here, and run this piece, just the name of the gene, DMTF1. Okay, so if we do that, and then that will do that loop for each of the consensus peaks. Go ahead and run this. It'll take probably a second to do. I've already done that. And so when we look at it now, our range of consensus peaks, each one of these is a G ranges, and it's now named. So that's handy. So it's better than having it 1 through whatever and having to remember which DNA binding protein is which. Okay, so now we have a named list of G ranges called consensus peaks. And let's take how we did before, and this is going to be a common strategy of just making a data frame and then starting to coarse it, add columns to it. Again, we can think of this sort of like an Excel sheet where we're going to turn it into more of a flat file, the way we think about it typically. So numpeaks df is something we're going to start to populate and add more properties to. We're going to say it's a data frame. One column will be named DBP, and it's going to be named after the consensus peaks that now have a name on it. So it'll be the name of the DBP. And the number of peaks will be, if you take the consensus peak, and it'll do the length of that and say how many peaks were called in each file. Okay, so let's run that and take a look at our numpeaks df. Okay, so we have the name of the DBP, which we set it to, and the length of the number of peaks. So we saw that a file will have 1, 2, 3, 4, 5, 6, 7, 8. It's just going to keep counting those until it knows the total number of peaks. And we can already see somebody only has 2 peaks. That might not be very helpful. And somebody has 144,000 peaks. So we can already see which DNA binding protein is covering the most real estate of the human genome. And that's in our numpeaks df there. We want to work with data where there's enough peaks to actually work with. Last year, we picked this number 250. We'll start with that here. We're going to do, you can do this exercise below to do some math to find out where the 25th quartile would be using an empirical cumulative distribution function. That's a bit more than what we want to do for right now, but if you want to sort of move ahead, that's one way to look at that, where you want to say not just an arbitrary number, but you say, oh, at the 25th percentile, I want to take out that data because there's not enough peaks in it. But we'll just set numpeaks threshold as a number, so that entry will just simply be a value here of 250. And then we're going to make a filtered consensus peaks of all the Chip-seq files that have more than 250 peaks. So we're going to take consensus peaks, we're going to index into it, into numpeaks data frame, number of peaks has to be greater than 250. So then that's going to reduce consensus peaks entry here down to the name, to the only the entries with more than 250 peaks in it. Okay, so let's run that. Okay, so now we have filtered consensus, and we can see now it's only 460 long. So if we look at that, it's going to have the same named G ranges, but now it's length 460, whereas before it was 483. Okay, so now let's start looking at some of this data. We're going to go over plotting in a couple more classes. For now, we're just going to focus on the aesthetics layer, and we'll go into much more about R plotting in the future. But just so we can start to analyze some of our data, we're going to do some basic plotting here. ggplot is worth searching and looking up. It's what it is. That's what we'll be plotting mostly with in R. And R is really handy to plot in, and we'll just start with the basics for now. So let's just see how many peaks each protein has. And so we can say that in our numpeaks df here, in our numpeaks df, we can see that we have the number of peaks and a bunch of DNA binding proteins. So let's plot that as a histogram, which the x-axis will be how many peaks are possible, and then the frequency on the y-axis. Okay, so if we do ggplot, and then it's going to want to know what to be plotting, and we're going to look into numPeaks df, and we're going to set the aesthetics layer, which is always done after this comma, of x equals number of peaks. We'll see later when we add a y-axis as well, and you can add more as we'll see in future classes. So really we're saying the x-axis to numPeaks, and then we're going to call a histogram from the geom line, which is the next input into our plotting, and saying like, what kind of plot do you want? We could have just done hist up here, but we'll run this way. Okay, and bins is just the number of breaks that's going to be on the x-axis. And here we're just picking 70 arbitrarily. And you can see what that is without having to do all this other fancy plotting really quick by just doing show as a base R function of G. And so now what we're seeing here is there's at least one DNA binding protein, as we saw before, that has over 150,000 peaks, but a lot of them have near zero. And that's why we want to cut off around 250. And we're looking at the numPeaks df before it's been filtered. And we'll clean that up in a second. Okay. The rest of this is just here if you want to see that. So for now, we're not going to go over all of this fancy stuff here. You can go ahead and try and run it. If you do, you're going to need to run that exercise we had up here. Asking what percentile. You'll have to make this threshold percentile value here in order to make this plot. Okay, but so now we can see already that there's a wide distribution with sort of the majority being somewhere around 50,000, under 50,000 peaks, and a few outliers that are 100,000 or more. Okay, so that's good. And so what we're going to want to do before we go too much further is these consensus peaks are now, or the filtered consensus peaks are now in our sort of environment, but they're not files. So let's make them into files before we go much further. So what we're going to do is a for loop to go through and take those G ranges in filtered consensus peaks now, so 460 G ranges, and it's going to run this function, again, calling a genomic ranges function, R track layer export now. So now it's going to export all those ranges that it's kind of keeping in the environment into an actual bed file. And R track layer export does have a parameter that you can tell it what file type it is. And we'll see that in a second. But for now, what this export function is going to do is run on the first file in consensus peaks, one of the 460, and it's going to paste the results filtered peaks as the file path to put it in. So the first thing is saying what is the file we're looking at. Now paste this file path, and now we're going to name this file. So the name of this file, we use names of filtered consensus peak I, which will be the name of the DNA binding protein that we just saw. And then we're going to append this text, and that's why it's in quotes. So we're going to say the DBP name underscore consensus underscore peaks underscore filter dot bed, and it's going to put it in this file path here. So when we run this, I'm not going to run it because I've already run it, but when you run it, it might take a little while. But it's going to export all those filter G ranges just back into bed files. So we have a nice list of consensus peak files that are filtered that we can see. What it'll look like is you'll go into your results and you'll now have a new folder called consensus peaks, which you should populate before you run this. And when we look at consensus peaks, this is where we'll be working from moving forward with these files. Okay, so now that we have a nice set of those files, we can go and still do some more analysis on those files that are in the environment already. So in numPeaks df, we have the number of peaks in there, but we might want to have something else, like how much of the genome did those peaks cover? So what if they were all really small peaks? then they wouldn't cover as much genome space, even if they had large numbers of peaks. So let's just take a look at that. And what we're doing here is sort of a tidyverse thing where we're saying, here's the data frame we're interested in, pipe it to filter, which we've seen. So we're going to filter the DBP name in the names of filtered consensus. So this is a really nice, tidy way of writing this, is saying I'm looking for this, the DBP name, in the names of filtered consensus peaks, which names can extract those and match. So let's take a look at that. And really, all we just did is set our num-peaks df now changed from 483 to 460. So we're going to compute off this list of G ranges, sorry, this data frame that we had before, but it was 483 before, and we want to move to just the filtered ones. So really what we did is we just changed num-peaks by filtering it to only the names that are in the consensus peaks. Okay, so now we have that data frame, which again, you can sort of think of as an Excel file. And now we can index into that data frame and make a new column. So this is where we're making a new column by saying, okay, now there's an index with this cash sign, and it's going to be called total peak length. Okay, and so to get the total peak length populated, we're going to use sapply. Again, we're going to use it on the filtered consensus peaks. So we're down to the 460. And we're going to run a really simple function that we don't even need brackets with, because it's this function x, which is just simply base our commands, which is sum the widths of x. So x is going to be all the peak widths there are, and then they're going to sum them up for that given file. And then it's going to populate that entry. So now every DNA binding protein is going to get the sum and width in each row until it runs out of rows in filtered consensus peaks. Okay, so let's run that. Cool. So now let's take a look at our num-peaks df. We see there's a new observation there, 3 variables instead of 2. And so before we had the number of peaks, and as you can imagine, the more peaks you have, the more genomic length you're going to cover. And notice we don't have the one with 2 peaks anymore. Everything is above 250 peaks. Okay, but you can see this guy has nearly more peaks, but covers less space. So these are probably more narrow peaks than these. So already some little bits of information you can start to figure out. But now let's plot that more globally and just ask, is there a linear trend that the more peaks you have, the more genome space you would cover? So again, now we're going to use an X and a Y. So in ggplot, we're going to tell what we want to plot in, num-peaks df. And then we're going to set the aesthetics layer, or the X and Y, or the axes. As X is going to be the number of peaks, and Y is going to be that second column we just made of the third column of total width covered. And then this is just saying label the plot by the each dot by its name, DBP. So let's run this. And again, don't worry about all this. We'll get into these layers that we'll add in R, but you can see that you essentially program figures, which is really powerful. Okay, all right, we already know now we see a very linear trend. We'll learn stats later in course on how to determine if this is significant. It's kind of bulky because we labeled each point, but we can see this ZNF 280 protein out here is our largest number of peaks and also covers the largest amount of the genome. H3K4 monomethyl is interesting. This is a histone modification that packages DNA. And this histone modification indicates that it's an enhancer region. And we'll discuss more of that later. But it's covering a lot of the genome. So it's interesting. We're already starting to see when you have 480 or 460 different DNA binding proteins, we see that they do all pretty much share one trend of the more peaks they have, the more genomic space they cover. Okay, so now let's find overlaps with GenCode. The previous video did that for promoters, and that's kind of what we're going to want to do. This is where I had mentioned as 166, but to load this GenCode GR, it might take a minute. I already have it loaded. And if you want to get more familiar with this, the previous video goes into much more detail of what's in here. And in that video, we also filtered GenCode because there's a lot in here. When we look at GenCode GR, we can see, if we look at it here, we can see this at element metadata and ranges we looked at before. So ranges is going to tell us the widths and starts, ends of features. And in element data, we can start to ask for very specific things like the name of the gene. We went into type to get exons, or gene, which is the gene body. And we're going to do some more of that now with our bigger data set. So load that in. And let's say we want to make a object that is just the long non-coding RNA and mRNA promoters. We saw in gene that there's all types of genes. There's pseudogenes, process genes, and we're just going to look for long non-coding RNA and mRNA promoters. And so get promoter regions is a function that we saw previously in G ranges where you use promoters. You can look up how we've just modified that slightly to get promoter regions so that we don't have to put in the plus 3 kb and minus 3 kb every time. So that's really all this is doing. Is G ranges has the promoters function, which is going to look for the start of the gene, and then it needs two parameters of how much space you want upstream and downstream. So we just clean that up, have a look in the util folder for get promoter regions, but it's really no different than what we went over last time. Get promoter regions is going to run essentially promoters on GenCode GR, get the start site. Biotype is a parameter we have in here. You could use these indexes to do the same thing if you want to change the name. But in our function, we made a biotype, which is going to essentially go into gene type and go in there and pull out. We have to tell it what to pull out, and we're going to pull out long non-coding RNA and protein coding there. And then once we index into here and grab those, we're going to export that into our results folder as a GTF, because we might want to use this often and not have to run the code every single time. But I will run this part here. And so now if we look in our environment, we have this lncRNA and mRNA promoters, G ranges. So now we know where the start and stops of all those genes are. I've already done this. Feel free to export it. We're going to be calling up this file a lot, but to be honest, this is a pretty quick function, and we could just rerun this line every time. So let's make another object of just long non-coding RNA promoters so we can compare long non-coding RNA and mRNA promoters. We're doing the same thing, running get promoters, get promoter regions function, which needs the file or the G ranges, and then the biotype is the same as just going into type into the element data. Just makes it a little shorter to run each time. So they're great. We have a lncRNA promoters, large G ranges, and there's about 16,849 of those. We can do the same for mRNA, where we're now just filtering on the biotype protein coding. And now we see mRNA promoters is a long range of about 19,000. So there's about 3,000 more mRNAs than long non-coding RNA promoters in GenCode. Okay, so now let's count the number of times a given DNA binding protein overlaps with a promoter. So in the previous video, we did that just for POL2 consensus peak, and we made the G ranges of the POL2 consensus peak, which we now have a list of, a bunch of them now, instead of just one, and also G ranges of just promoters. And then we did find overlaps between the two of them. So that's what our CountPeaks per feature is doing. We essentially, the same way we cleaned up the promoters function in G ranges, CountPeaks per feature is essentially going to do find overlaps. And again, you can look this up in the util documentation under intersect functions. Look for that. So CountPeaks per feature is, we can just see essentially we're going to do count overlaps, which is doing finding overlaps, and we're subcalling different functions. We went through the genomic ranges, names of these things. Feel free to take a deeper dive into here. They're not very long, and we're just formatting it to provide more of what we want for this class, so we don't have to put in as much information each time. Okay, so promoter peak counts is going to use that function, which is essentially just going to do the find overlaps that we did in the last video over and over again for lncRNA promoters in the filtered consensus peaks, and the type we're going to want out is counts. In the function, this is going to make it, we can make it a matrix or counts. And for now, we're just going to make it counts. But again, that's in this, in here, where there's a parameter that you can the type is going to either be counts or occurrence. And we're using counts right now because we just want to know the numbers. Okay, so let's do that. Let's get promoter peak counts. Essentially just doing find overlaps with the promoters using functions that we've just sort of added what we want into the genomic ranges, so we don't have to do it every time. Okay, this is going to take a minute. As you can imagine, it's going through a lot of data. Okay, so that probably took a minute to run, but now we have the promoter peak counts variable here. It's a large matrix. We probably don't want to open that. Oh, too late. I'm going to close it because it might be too big to open. So you got to remember, it's 460 proteins by all the promoters that they sort of intersected with. But we can add this number right into our numpeaks df and continue to build on it, where it's currently at 3 variables. We're going to make a new column called peaks overlapping promoters. And what row sums is a base R function that's going to go through each line here and sum each row for the number of peaks. Remember how we had the name with find overlaps will give you the name _1, 2, 3, 4, 5, 6, all the way down. And it's just going to go through and sum the number of counts there are. And so if we do that, we can see our numpeaks data frame now has peaks overlapping promoters. So we can now see if the majority of the peaks are overlapping promoters, or who might prefer promoters versus not preferring promoters. And so let's take a look at that. And now actually we'll make 2 new entries into numpeaks data frame, same way, by indexing with this cache sign. We'll make one for lncRNA promoters and one for mRNA promoters, so we can plot them separately and just sort of compare the trends. Okay, and here we are row summing, and we have to tell it that we're looking for long non-coding RNAs here. So we'll do row sums on promoter peak counts, but we're going to index into all the rows, but only where the long non-coding promoters gene ID is. So this will separate out only those ones, and then we'll do the same thing for mRNAs in gene ID. Okay, so we're really just indexing and summing the rows for those biotypes or gene types in gen code terms. Okay, so we'll do that, and mRNAs. Okay, so now we have in our numpeaks df even more entries, and we can see now the total number of peaks overlapping, and then how many are from a lncRNA and how many are from an mRNA. Okay, we're going to plot this. We'll plot the number of peaks on the x-axis, and the y-axis will be the number of peaks overlapping promoters. So if you might expect some linear trend that the more number of peaks you have, the more likely you are to overlap a promoter. So let's take a look at that. Again, don't worry too much about the rest of this. We'll go over this in the R plotting class. Okay, so what we see here is, so this is the total peaks overlapping promoters, is the total number of peaks. We haven't broken it down by lncRNA or mRNA yet. And the x-axis is the peak number. And so what we see is what we'd expect is a linear trend here, where the more peaks you have, the more times you would overlap a promoter. And in fact, in the last class, that's what we saw as a near perfect linear trend here, But with this larger data set, we're seeing these values out here, which are indicating that they have more peaks that don't overlap promoters. And that's quite interesting. And we'll look at that further in class, because that is a different trend than what we saw last year with 195 DNA binding proteins. Okay, so now let's look at this by the two different types, or let's look at the gene bodies. So right now what we've done is we've done the promoters with our find overlaps, or all that could have been done with find overlaps and promoters functions. In G ranges, we just made our own functions to add our own parameters. So for lncRNA, mRNA gene bodies, we can also not just overlap the promoter, but the entire gene body. And we saw before in type gene, then we only get things that are genes, and they're not pseudogenes or other types of things. And that gives us just one entry for a given gene name. And then the type we saw is where we can separate out lncRNA and protein coding genes. So here we can just index our current GenCode GR and get the gene filter down to genes in there, and is a new thing we're seeing. So we're running two indexings in one index here by these square brackets. So we're going to GenCode GR type, and we want gene in there, which we've seen in this and before, the class before. And then the gene type is lncRNA or protein coding gene. So this will give us the start and stops of those gene bodies in there. And so now what we can do is count peaks per feature. This is why we made this function. So if you don't want promoters, you don't have to call up a different function. Now we can just say whatever feature you want in the human genome, just use this function and it'll do find overlaps and other parameters in there. You can read in the util folder. But as you see, as we go in class, this will make it much easier when we start to intersect lots of different things. So we're going to just count the number of times is the count peaks per feature function is going to do the overlaps. And then we're going to overlap mRNA, lncRNA, and mRNA gene bodies. And we're going to do that in the filtered consensus peaks and types. Again, we had to say if it was a matrix or counts. And we're just going to count for now how many times there was an overlap. So basically find overlaps gives us that index of where peaks overlapped, and count peaks per feature is going to keep that and then provide the total number of times a feature overlapped a given DNA binding protein. Okay, we'll run that. That might take a second as well because we are permuting over a lot of DNA binding proteins. Okay, so that is now done running. And so now what we have is the number of overlaps, same way we had with promoters. It looks very similar to what we did up here. We have the number of counts over gene bodies. This is just as a helpful stopgap here that we want to make sure when we do that, when we compare the gene body counts, that it's the same as the number of peaks as the promoter. So we should expect an account of entry the same length for gene body counts as we would for promoter counts, because there's only one associated with each gene. There are ways that that could have been mistaken, and then that would be a big problem. So this is just making sure if that's not an issue, we can keep going. So it's not an issue, and stop if not is a base R function to say sort of check your files and make sure they're the right lengths. We'll see why that's more important later in the class. And now we're gonna do the same thing we did with the promoter, is make a new index into our num-peaks df of peaks overlapping gene body instead of promoters, and using the same row sums function to go through what's now being stored as the gene body overlap indices and just counting them rather than producing a matrix, which we'll do right after this. Okay, so now we're gonna add this entry. Cool, so now we have promoters and gene bodies. And let's do the same plot we just did for promoters, but now with peaks overlapping gene bodies that we've now defined. Okay, so we'll get a plot like we have one on the right here. Wow, okay, so that's much more linear. That's quite very similar to what we saw last year, that despite many of those in the previous plot having off linear functions, meaning that they're outside of promoters, Well, where they pretty much are, if they're outside of a promoter, is in a gene body. This is a very nice linear relationship. And that's quite interesting because what we saw in the previous video is that gene bodies don't cover much of the total human genome. So these DNA binding proteins really know to go near genes, both lncRNA and mRNA genes here. Okay, so this is really nice. We have all these things, but now that we've done this, a few of these functions, we don't want to run them again. So we're going to make some flat files that we can use in the future. And so here we had type before. So we did CountPeaks per feature, and we did lncRNA promoters in the filtered consensus. And this will give us in both promoters. So both lncRNA and mRNA promoters, we're going to count the peaks per feature, and then only looking in the filtered consensus peaks, and the type is occurrence. And this is going to give us a large matrix back that we can use right table for promoter peak occurrence, which we just created. I'm not going to run it because count peaks per feature takes so long, and I've already made this file. But we can do right table to make this output file, so we don't have to run any of this again, and we'll have num peaks df kind of in a sort of what you're thinking of as a more of a matrix of rows of promoters and features across the matrix, in the columns of the matrix. And so we'll want that in that form. I'm not going to do it now. Go ahead and run that. Again, this is just going to check to make sure that the right number of lines are there and that one column doesn't have more lines. And then this is what we're more used to seeing. So this is going to be something we can compute on because it's a matrix. We'll get into why that's important, but this will be a different way of computing the same kind of information of how many DNA binding proteins are on a given promoter in future classes. However, let's also do it as the more way we would think about it, as a data frame or more like an Excel sheet. So we have all this information in num peaks df. We have the gene ID, or we could get all of this data from GenCode, and where we can say we've made all these objects, and now we're going to get the parts out of them that we want. For the gene ID, we just do the call names of the promoter peak occurrence. Remember how we added the name to the list of G ranges? So now we can just get that from that column. The gene name can come from the lncRNA promoters file, gene name, where we added that one. Gene type, same thing. Chromosome, now we're seeing, we're using seq names. Remember how seq names, if we did seq names, GR or GenCode, we got the chromosomes. And so this is gonna get us all of the chromosomes that are in seq names, which we've looked at before. Again, that's a really handy feature of genomic ranges to know that it can just know those things by seq names. We're making the promoters of these. We're going to set the coordinates of the promoters that count peaks per feature used in a version of the promoters genomic ranges function. So that's just saying where our promoters start and stop. The strand we can grab from again, all this metadata that in these G ranges, we have all this metadata that we can always get from. And so we can get this, the start and the strand in here and other things that we can index. Okay, so we'll grab the strand, and then the number of DBPs, we'll call sums the promoter peak occurrence. We kind of already did that above, but this will tell us the number of DNA binding proteins at each promoter. Okay, so let's run this. It might take quick second. Not run this. So I do have to run this, and you might want to pause the video. This might take a second. Okay, so that finished running. And just a little background of why that was actually important to run is this occurrence matrix is going to tell us for each row is kind of a promoter. And so it's going to tell us, and each column is going to be a DNA binding protein, or 460 entries. And so it's like 60,000 by 460. And now what we can do is things like row sums across there. and easily grab the information of that we're still missing, which is how many DNA binding proteins are bound to each promoter. So in that matrix occurrence there, the row sum would be the number of DNA binding proteins for that promoter, that single promoter. And what we're trying to look for is like a promoter that has all 483, or who's got the most amount of DNA binding proteins on their promoter. So this will help us. This promoter peak occurrence is something we're going to need to call for the call names for the DBP. The name is the names of the matrix, so the columns, and then the promoters are the rows, and we can index into other objects we made above. So take your time and go through this a little more slowly, especially this chromosome is kind of cool, just doing the seq names that we've seen before. And then we will make this peak occurrence data frame, which will be kind of nice because it'll have all of our information in more of a way we're used to kind of seeing data. So this is called peak occurrence DF. See here, and look at this nice table we have now. So we have each row in that matrix was a promoter. We knew the column was the gene name, and we'll have a unique entry for each gene. The gene type, so we now have lncRNAs and protein coding genes mixed, the chromosome, where the 3 kb up point is, what strand the gene is on. We saw that in G ranges. We're going to kind of want that when we start looking deeper at gene expression, and then the number of DBPs bound at that promoter is here. So this is really handy, and we're going to keep using this table as we do more and more analyses. We'll just keep adding more and more features to each promoter and keep moving along this way, where we'll have an occurrence matrix to count stuff, and then we'll flatten it into a data frame as we just did here. And you can write this out. Again, I've already done it, so feel free to do that, because now that's a handy data table to have already. So let's look at what the density of binding is at promoters. So we have this peak occurrence data frame now that tells us the number of DBPs bound on a promoter here, number of DBPs bound. Okay, so let's plot that and just see what that looks like across all of them. So we're going to use ggplot to go into the peak occurrence data frame that we just made, and just set the x-axis to number of DBPs. This geom layer is the next layer we'll get into, is gonna give us a density plot. So we're gonna look at density instead of histogram that we looked at previously. And so let's just run this and see what we see. Okay, cool. So what we can see is a vast majority of the promoters are bound by at least one factor, because this black line is not starting at 0. And we can see, as we saw last year, we never saw more than 100 DNA binding proteins at a promoter. There was an asymptote there. But because we have this bigger data set, we can see the same trend we saw last year with a big drop at around 100. But what we didn't have the ability to see last year is this extra bump, where there's over 300 DNA binding proteins all on the same promoter. And so we'll start to look at that and see what's going on in there. So that's kind of cool. That's already a new result from last year's class. And so if we want to just look at a few of these features, such as unbound promoters, we could do this here, where we go into this peak occurrence data frame made just above, and we filter it. And we can filter into the index. So we're going into peak occurrence, numbers of DNA binding proteins less than 1. So how many have 0 DNA binding protein? And now we can see here we have this unbound promoters variable with 6,720 promoters that don't have any DNA binding proteins. And sometimes I like to just check on these things. So that's about 10%. That's not too bad. But that's a lot of DNA binding activity in the genome. But we could look at this really quick by clicking on our promoter peak occurrence data frame, is what we just filtered. And what we can see here is there are many promoters that don't have any DNA binding proteins. And we know it's 6,720. So we're at line 3,000 right now. So if we scroll to line this is painful, I apologize, but if we scroll to line 6,000, we should end up with promoters that start to have values because I sorted it on unbound over there. So we'll try and get to 6,000 as fast as possible here. We're getting there. But it is good to spot check like this, as manual as it is. I'm sure there's a fancier way of doing this, but we got to get to 6,720, and we should start seeing numbers. We're just about there, and here we are, 6,720. We should start seeing numbers, and we do right there. Okay, so cool. This makes sense. That's a lot of promoters, 6,000. And the 7 observations is the number of columns that we have in our NumPeaks DF, the 7 variables here. Okay, great. So we can write those out in a CSV here, and we can just filter them out from the rest of our analyses. Okay, so now let's see if a DNA binding protein never binds a promoter. And so that would be rare. That it means it would never find a promoter in all of this data. And so we can go through this code here and to get overlapping peaks between lncRNA, all the promoters, and our filtered consensus peak file. The number of overlaps we can get by just simply doing an sapply on this DBP promoter overlap we just made up here, and just call its length. And so that's the number of overlaps there would be, and we can see the min and so on. So let's do that real quick. Okay, it's run, and then so we can see the number of overlaps by just going across the length of that object. And what we can see is num overlaps. Should be a value here. And there's our number of overlaps. And then let's say, what's the minimum number of overlaps? 54. Okay, so no DNA binding protein has less than 54 overlaps with that. And we can see the summary that the max is a gene that has 27,000 overlaps with promoters. So it's essentially binding to half of the promoters, whereas the mean is about 8,500 promoters bound, which is close to what we saw to poll 2 in the previous exercise. So you can go over this a little bit more. It's just really asking details in our file that we already made. And now what you can do is try and do some of these plots where we separate out lncRNAs and mRNAs. We've left this as a class exercise as we've gone through quite a bit today. Again, please go back over this. We were going over this kind of fast just to get the concepts out there, but there is plenty of documentation to go back through. Okay, and this can be an exercise here where you just look at this, what's going on here. It's a lot. There's your mutate function. But try and think through this and see how we can plot separate the long non-coding RNAs and mRNAs separately using this code. And we'll go over a lot of this in the R plotting video coming up. So in the meantime, code light. Thank you.